Hey everyone, I hope you're all having a restful, though probably packed 3-day weekend. I apologize, the Camtasia site wasn't working over the weekend, and now we're on a YouTube video, so I hope this worked out for everybody. And welcome to Anatomy Lab Week 7, where we get to take a look at another vital physiological component, muscle physiology. This week we'll get comfy with nervous tissue and the nervous system, as well as work to understand reflexes. The responsibility of our nervous system is to coordinate voluntary and involuntary actions. The first division we'll notice is an anatomical one, the separation of the central nervous system and the peripheral nervous system. The central nervous system is one that includes the brain and spinal cord, both of which are protected by bone of the skull and vertebral column. Its responsibility is to integrate, process, and coordinate sensory data and motor commands. The peripheral nervous system consists of all nerves that conduct impulses or send signals towards or away from the central nervous system. These two systems communicate rapidly to coordinate bodily actions in response to environmental changes or stimulants. The peripheral nervous system is further divided by function. Whether the nerve is leading towards or away from the central nervous system determines its classification. An afferent nerve is one that leads towards the central nervous system. These are sensory nerves that carry information from receptors in peripheral tissues and organs. The nerve reaches the central nervous system, where it is processed and a command is formed. This motor command is relayed to an efferent nerve that is leading away from the central nervous system. Think of afferent nerves as accessing the central nervous system and efferent nerves as exiting it. There is yet another division among efferent nerves, the autonomic nervous system and the somatic nervous system. The autonomic nervous system exerts control over involuntary muscle tissue, such as cardiac and smooth muscle, while the somatic nervous system exerts control over skeletal muscle. We'll see later on how skeletal muscle can actually have a subconscious involuntary function in addition to the voluntary control of bodily movement. Remembering the defining characteristics of each of these muscle types will be crucial to determining which division of the peripheral nervous system controls it. Here we'll take a closer look at a neuron, a nerve cell that conducts electrical impulses. There are anatomical features of this neuron that are important to point out and spend some time studying. Dendrites are the slender branching processes that extend from the soma. They're responsible for receiving neurotransmitters from an adjacent neuron and pass this information to the soma or cell body. The axon hillock is the thick cone-like emergence from the soma and has a very high concentration of voltage-gated sodium channels. This is considered to be the place where an action potential is initiated. Directly following the axon hillock is the axon, which is where the message or action potential is carried away from the soma and toward terminal boutons, which in this case might be forming a neuromuscular junction on a muscle cell. Remember that a muscle cell is the multinucleated striated fiber, and many of these make up one muscle fascicle. The axon is often covered in a myelin sheath, which, as we'll see later, will aid in propagating action potentials. When referring to a bundle of axons, it's appropriate to call them nerves until they enter the central nervous system, at which point they're actually tracts. They send the same type of information and in the same direction, either sensory information from the spinal cord to the brain in ascending tracts, or motor commands from the brain to the spinal cord in descending tracts. A nerve itself is composed of layers that are comparable to those of skeletal muscles. The epineurium is the most superficial layer made up of dense collagen fibers. The interfascicular epineurium refers to the extension of the epineurium towards the perineurium. The perineurium divides the nerve into bundles of axons, also referred to as fascicles. Endoneurium surrounds the individual myelinated axon. Myelin insulates the axon in concentric layers of plasma membrane. This insulation increases the speed or conduction of an action potential down the axon. The myelin associated with axons in the peripheral nervous system is called a Schwann cell that contains a single nucleus. One Schwann cell makes up one myelin sheath, separated by nodes of Ranvier. On the other hand, the myelin associated with the central nervous system takes shape in the form of oligodendrocytes. Each oligodendrocyte, also containing a single nucleus, is capable of myelinating several axons. This nucleus is not visible in the myelin. Think of the oligodendrocyte as an octopus whose tentacles reach multiple axons while still being one cell. Neurons are classified in many ways, one of which being their anatomical structure or number of processes. A unipolar neuron is one in which the cell body lies to one side and the dendrites and axons are essentially continuous. Most sensory neurons fall into this class. In the spinal cord cross-section, we can see the dorsal root ganglion, which is where we find a unipolar cell. Upon taking a closer look, we're able to make out the individual nucleated cell body. The next type of nerve is bipolar. This means that the cell body lays in between two distinct portions, dendrite processes and the axon. These are very small and are found in olfactory and optical sensory tracts. This week we'll look at a slide of the retina and be able to see neat rows of bipolar neurons as well as ganglion cells that are receiving the visual information and the nuclei of rods and cones. Multipolar neurons are the ones found most abundantly and have multiple dendrites leading to a cell body with a single axon. Most motor neurons fall under this classification and they are the most common type within the central nervous system. This week in lab, we'll be looking at a spinal cord smear and observe the different components of motor neurons. Something that we're all relatively comfortable with yet may not completely understand are reflexes. Once the process is learned, we'll see that a nice story is being told here. Reflexes are rapid and involuntary responses to stimuli. When a receptor is stimulated and activated, a signal travels by way of an afferent neuron towards the spinal cord where it is processed and can either pass through an interneuron or association neuron before exiting the spinal cord, or it can be directly sent out through an efferent neuron. Notice that the brain does not dictate this motor action potential, but the sensation is relayed to it. The efferent nerve sends this signal to the effector, thereby eliciting an appropriate response. And again, this effector would be a muscle, and the efferent neuron would be creating the neuromuscular junction on the muscle, the single muscle cell. In lab, we'll be looking at three different types of reflexes, the first of which being the stretch reflex. A stretch reflex is one where the muscle spindle within the muscle serves as our receptor, and a muscle is the effector. As shown in the diagram above, tapping the tendon with a mallet will stretch the muscle spindles within the attached muscle. This stretch is detected and the response is sent back via the reflex arc, and skeletal muscle is involuntarily contracted. This includes the patellar or knee-jerk reflex that most are familiar with, along with the biceps, Achilles, and triceps reflex. Take a look at the links posted on Blackboard in order to better understand what these reflexes look like when properly performed. This can be a diagnostic tool for suspected nerve damage. In a cutaneous reflex, muscles serve as the effectors and involuntarily contract following a stimulation of the receptor or skin. This week we'll practice the abdominal and plantar reflexes. In a normal adult plantar reflex, the toes flex or curl. In infants, the toes move apart in what is called the Babinski response. This is due to the lack of myelination of the nerves. If a Babinski response is observed in adults, it would have a pathological indication. The Blackboard videos show the effects of these reflexes when properly performed. Cranial reflexes are controlled by one of the cranial nerves and are processed in the brain. These reflexes are important because they can give an indication of the condition of specific cranial nerves. In lab, we will only be practicing the pupillary reflex, which gives an idea of the functionality of cranial nerve 2, the optic nerve. You will shine a light over the eye of a partner and observe the change in diameter of the pupil. In the corneal reflex, we would have one person simulate an object flying towards the eye. Take a second and think about what would happen and why this might be an advantageous reflex. The videos give examples of functional cranial and pupillary reflexes. As the name implies, the neuromuscular junction is where the nervous and muscular system communicate. A muscle fiber or cell is controlled by a single axon of a motor neuron that forms a synaptic terminal at the level of the sarcolemma. Here, a neurotransmitter is released from synaptic vesicles and triggers an influx of sodium ions at the postsynaptic membrane, which will result in an action potential and therefore calcium release down to the level of the thin filaments or actin. This week in lab, we're going to be looking at this connection between the nervous and muscular system. Notice in this image that a motor nerve is composed of multiple neurons that can innervate or control a large number of muscle fibers. A motor unit is made up of all of the muscle fibers that are controlled by a single neuron. So in this case, all of the coral muscle fibers would be part of one motor unit. Recruitment is referring to the rise in tension of the muscle as the number of active motor units increases. This week we'll be recording an electromyogram, or an EMG. An EMG measures the electrical activity or impulses generated by muscle cells. An EMG does not measure the strength of the contraction, but it can be indicative of muscle use. The hand dynamometer will be used in order to measure the force at which the muscle is being contracted, and in doing so will give us the ability to compare EMGs at different grip magnitudes. Notice in this graph that the motor recruitment is indicated on the y-axis and time or duration on the x-axis. When applying the electrodes this week, first place the green electrode near your wrist, then the black and red electrodes on the forearm and antecubital regions. Remember, green is a ground electrode. This week in lab, please keep your phones away from the iWork station or just throw it in airplane mode. The equipment is really sensitive to electrical signals and phones can throw off our readings, and that is no good, so please be aware of that this week. And also don't forget about the iWorks pre-lab that is due at the beginning of lab, along with the pages in your lab manual. Next week you'll have to turn in a graph of the iWorks information that you have collected, but we'll also be stamping the iWorks handout that you can print off of Blackboard. That'll have some tables in it that you'll need to fill out at the iWorks station. Good luck studying, and thanks for putting up with this awesome YouTube video instead of Camtasia, and please enjoy the rest of your 3-day weekend.